In the previous lecture, we explored the electrical conduction of the heart. In this lecture, we will discuss the electrophysiology of the heart. This will give us an understanding as to how the EKG gets its information. We will review the mechanics of myocardial contraction, electrical potential, and the phases of myocyte activation. Electrolytes are the means by which cells develop their electrical activity. An imbalance of electrolytes can have life-threatening consequences. Therefore, understanding the effects of the electrolytes and how they may affect the EKG can potentially save a patient's life. The semipermeable membrane of the myocyte results in different intracellular and extracellular ion concentrations. As seen in this image, the inside of the cell has a greater potassium concentration. So inside the cell, which is those labeled here in red, that's potassium, while the outside of the cell has a greater sodium concentration. So here's our sodium, and you notice it's much greater on the outside of the cell. There's also a greater concentration of calcium outside the cell compared to the inside. The higher positive charge outside the cell causes a relatively negative charge inside the cell. So it's more positive compared to the inside. This difference in electrical charge between the inside and outside of the cell is known as electrical potential. Due to the semi-permeability of the cell membrane, some ions can leak in and out of the cell. Because of the greater concentrations of potassium inside the cell, it has a greater tendency to move down its concentration to exit the cell. On the other hand, because sodium has a greater concentration outside the cell, its natural tendency would be to move down its concentration gradient and thus enter the cell. So the sodium outside will move down its gradient inside to the inside of the cell. Note the sodium and potassium ATPase pumps in the myocyte membranes, which are these colored in blue here. These are in place in order to maintain an electrical potential. These pumps push ions against their natural tendency through an active, energy-dependent process. They will expel 3 sodium ions out of the cell and bring 2 potassium ions into the cell. This goes against their concentration gradients and therefore requires energy to do so. The result of this process maintains a greater number of positive charges outside the cell compared to the inside, and an electrical potential of about -70 to -90 millivolts in a resting myocyte. Let's take a moment to draw out and review what we just discussed. Let's imagine here that this is our myocyte. So we'll label this our myocyte, and this outside, the extracellular fluid, and here the intracellular fluid. Remember, we discussed previously that there's a greater concentration of potassium inside the cell compared to and a greater concentration of sodium, as well as a greater concentration of calcium outside of the cell. The higher positive charge outside the cell causes a relatively negative charge inside the cell. The difference in electrical charge between the inside and outside of the cell, this here, we can call, is what results in an electrical potential. And we said that the myocyte has a semi-permeable membrane. And what this allows is for some ions to leak in, such as the sodium, down its concentration gradient, and some potassium to leak out down its concentration gradient. We also said that there were sodium ATPase pumps in the myocyte membrane that are in place in order to maintain this electrical potential we discussed. So we can call these our sodium potassium ATPase pumps. And these pumps, we said earlier, push ions against their natural tendency through an active, energy-dependent process. They will expel 3 sodium to the outside of the cell and bring in 2 potassium to the cell. This goes against their concentration gradients, as we said that there's a greater potassium inside the cell, and therefore requires energy to do so. And this results in a process that maintains a greater number of positive charge outside the cell and compared to the inside of the cell, and that results in the electrical potential that we mentioned before of about -70 to -90 millivolts in a resting myocyte.